From beautiful Sydney, the city of Opera House, Darling Harbour, Blue Mountains and beautiful beaches, this is ICU Primary Prepcast, a monthly podcast about physiology and pharmacology in intensive care.
Hi, I'm Maddi.
Hi, I'm Swapnil. And we are back again with another episode of ICU Physiology. Maddi, it's been kind of interesting 2 months where we all been in lockdown and COVID-19 has impacted everybody's life, I guess. How are you feeling after this lockdown has finished?
It's pretty good. I think we've been lucky in that it hasn't affected us too much. Like we still have a job, still can see people at work. And I think Australia's been minimally impacted from a health point of view. But yeah, I think as medical professionals, we've been quite lucky.
That's right. And massive thanks to the entire community for following the strict guidelines with social isolation and social distancing. I think the community did a wonderful job to protect us as a healthcare workers, I guess.
Yeah, definitely.
So Maddi, today we are going to discuss about immunology. So let's start with the first question. Can you please outline the components involved in innate immunity?
Innate immunity is the first line of defense of the body. It is non-specific and consists of lifelong immune mechanisms that do not require prior exposure to antigens and are not modified by repeated exposure to a pathogen. The innate immune system consists of physiochemical barriers, soluble proteins, and cellular defense mechanisms. So first, thinking about the physiochemical barriers, these include physical barriers and chemical barriers. Physical barriers are things like the skin, where the epidermis provides a layer of physical defense and is acidic. And this layer also sheds periodically, which removes some of the microbes. It also includes mucous membranes, hair within the nostrils, which filter microbes, dust, and pollutants, and it can expel them via sneezing. Cilia, which lines the upper respiratory tract and can trap foreign particles and remove them through mucociliary clearance and coughing. And the high flow rates of things like urine, saliva, tears, and biliary secretions, which can also remove foreign material. Chemical barriers include sebum, which provides a protective layer on the skin and is bactericidal. Lysozymes in tears, sweat, and saliva, which breaks bonds between components of the bacterial cell wall, resulting in cell lysis. Gastric juice, which is highly acidic and can destroy this bacteria. Saliva, which facilitates the clearance of microorganisms within the oral cavity and helps maintain the normal microbiota within the mouth. And the normal bacterial flora, which colonizes various parts of the body and competes with infective microorganisms. The next part of the innate immune system are soluble proteins, which include complement, acute phase proteins, and cytokines. Complement is an enzyme system consisting of more than 30 glycoproteins. They can be activated by the classical pathway, which is triggered by immune complexes, the alternative pathway, which is triggered by various viruses, bacteria, fungi, and tumor cells, or the lectin pathway, which is activated when lectin binds mannose groups in bacteria. Activation of these proteins lead to proteolytic cascades, which destroy invading organisms by opsonization, chemotaxis, and cell lysis by the insertion of perforins into cell membranes. Complement also plays a central role in the inflammatory process. It removes immune complexes and serves as a bridge to the adaptive immune response by activating B cells and regulatory T cells. Acute phase proteins include things such as CRP and alpha-1 antitrypsin and are mainly synthesized by the liver. They are also involved in opsonization and regulation of inflammatory mediators during the acute phase response, and they facilitate cellular defense and repair. Cytokines are proteins that mediate many of the cellular responses involved in innate immunity. They are produced in response to an antigen and function as chemical messengers to activate and regulate the immune response. The final part of the innate immune system are cellular defense mechanisms, which consist of leukocytes, mast cells, and natural killer cells. Firstly, thinking about leukocytes, they are cells formed in bone marrow and lymphoid tissue and transported in the blood to areas of inflammation. Neutrophils, eosinophils, basophils, and monocytes protect the body by release of cytokines and inflammatory mediators and by phagocytosis. Neutrophils are the first cell line of defense in the innate immune response and circulate within the bloodstream. They are attracted to areas of inflammation by chemotaxis. They exert antimicrobial action through degranulation and release of reactive oxygen species and cytotoxic components. They can also phagocytose pathogens. Eosinophils also phagocytose microorganisms and release cytokines that produce inflammation and kill organisms. They are less active against bacteria compared to neutrophils, but are increased in parasitic diseases and allergic responses. Basophils are unable to ingest foreign cells, but they contain granules filled with histamine and participate in immediate type hypersensitivity reactions. They are important for protection against helminth infections. And finally, monocytes and macrophages enter the blood from the bone marrow and circulate for approximately 72 hours, following which they migrate to tissues and differentiate into macrophages. They are activated by cytokines and bacterial endotoxins. Their functions include phagocytosis, breakdown of damaged tissues of cells, release of cytokines which recruit other immune cells, and antigen presentation to helper T cells. They are especially important in the defense against intracellular pathogens, mycobacteria, parasites, and fungi. Mast cells are heavily granulated cells of the connective tissues, which contain proteoglycans, histamine, and proteases. When they are activated, degranulation occurs, which creates an inflammatory cascade. They are also involved in enhancing angiogenesis and are especially important in the homeostasis of the commensal bacteria of the gastrointestinal tract. They are also involved in acquired immunity by expression of high-affinity IgE receptors and are involved in allergic reactions. And finally, natural killer cells are present in the blood, spleen, and liver and contain granules with distractive enzymes and chemicals. They identify cells expressing low levels of major histocompatibility complex class 1, such as tumour or virus-infected cells, and bind to them. They can then kill these cells through release of cytokines and insertion of perforins.
Thanks, Maddi. So one of the key functions of our immune system is to protect our body from foreign elements, including the viruses, bacteria, fungi, as you mentioned. But also at the same time, it's very important that our immune system does not attack our own cells. So one of the key unit of immune system is major histocompatibility complex, that is MHC complex. So can you tell us a bit more about this MHC complex?
Sure. So when antigens are engulfed by human cells, they are transported to the surface of these cells and are attached to proteins to be presented to the body's defense mechanisms. These proteins are called major histocompatibility complex proteins. So the major histocompatibility complex is a group of genes located on chromosome 6 that code for these cell surface proteins, which help the immune system distinguish foreign proteins from its own proteins. The MHC is polygenic and polymorphic, thus making it difficult for pathogens to evade the host immune response. There are two important classes of MHC, class 1 and class 2. Class 1 proteins are expressed on all nucleated cells and present antigens to cytotoxic T lymphocytes, which then kill these infected cells. Class 2 proteins are only found on B lymphocytes, macrophages, and other professional antigen-presenting cells, and these present antigens to the helper T cells. Class 1 MHC proteins are especially important for the detection of intracellular pathogens such as viruses, while Class 2 MHC proteins are particularly important for the detection of extracellular antigens, such as bacteria. There is also a Class 3 MHC, which encodes for components of the complement system and other inflammatory molecules. MHC is also called human leukocyte antigens and is a crucial part of organ transplantations. There are three major MHC class 1 genes known as HLA-A, B, and C, and three major MHC class 2 genes, as well as several minor genes. The closer the match between the donor HLA complex and the recipient's HLA complex, the lower the risk of organ rejection.
That's right. So this HLA plays a very important role in the whole organ donation. And that's why matching the blood group along with HLA compatibility is really, really important. Now, can you please briefly describe the function of T cells and B cells?
So both T and B cells are lymphocytes that are derived from multipotent hemopoietic stem cells within the bone marrow, and they are a vital component of the acquired immune system. T progenitor cells migrate from the bone marrow to the thymus where they mature. They cannot bind to antigens directly, but need to have broken down peptides of the antigen presented to them by antigen-presenting cells. T cells are involved in cell-mediated immunity. There are 4 main types of T cells: helper T cells, cytotoxic T cells, regulatory T cells, and memory T cells. The helper T cells make up the majority, or about 2/3 of the T cell population, and they express CD4 receptors. They produce cytokines, activate macrophages, and stimulate B lymphocytes to form plasma cells and antibodies. Helper T cells bind to MHC class II molecules. Cytotoxic T cells express CD8 receptors. They are responsible for removing pathogens and infected host cells. Regulatory T cells express CD4 and CD25 receptors. They help to modulate the immune response, prevent autoimmune disease, and prevent cytotoxic cells from causing an excessive immune reaction. And finally, memory T cells are the last group. Following an infection, antigen-specific memory T cells are formed. These are important because they can quickly expand their colony upon re-exposure to an antigen, thus providing a quick and effective immune response. B cells are involved in the humoral immune response. They can be activated directly by an antigen, unlike T cells. They can also be activated by T cells. When a B cell encounters its matching antigen, it engulfs it and displays fragments on MHC class II molecules. This attracts helper T cells, which secrete cytokines and helps the B cell clone proliferate, producing plasma cells. Plasma cells then secrete antibodies to specific antigens, which can directly attack antigens and also activate the complement system. Some of the B cells also differentiate into memory B cells, which become activated if there is a reinfection by the same antigen. This helps to launch a rapid response against that antigen in the future.
Thanks, Maddi. So one of the most important component of entire immune system is antibody formation or immunoglobulins. So can you please describe what are immunoglobulins and how they function?
So immunoglobulins are also known as antibodies, as you said, and they are large Y-shaped proteins which are produced by plasma cells and are a vital part of the immune system. They can directly attack invaders by agglutination and lysis, or they can indirectly attack by activation of the complement system. Immunoglobulins consist of 2 heavy chains and 2 light chains, which are joined to form a Y-shaped protein. Both the heavy and the light chains have a constant and a variable region. The constant regions determine some of the biological properties of the antibody, and they are divided into 5 major classes: IgG, IgM, IgA, IgD, and IgE, depending on the constant region of this heavy chain. The variable region contains the antigen binding site and mediates antigen recognition. This gives the antibody its specificity for an antigen and provides diversity, allowing the immune system to recognize a wide variety of antigens. Immunoglobulins occur in 2 major forms: soluble antibodies, which are in the blood, and membrane-bound antibodies, which are attached to the surface of B cells and otherwise known as B cell receptors. The membrane-bound immunoglobulins detect antigens and facilitates the activation of B cells and subsequent differentiation into plasma or memory B cells. Now, we'll go a bit into detail about the 5 different immunoglobulins. So IgG is the predominant immunoglobulin class which is present in human serum, comprising about 70% of the total population. It is the only class that can cross the placenta and thus can provide passive immunity to the fetus. It is the primary immunoglobulin involved in the secondary immune response. IgG activates the complement system, opsonizes bacteria, and neutralizes toxins and viruses. IgA is the second most populous class and comprises about 15% of total immunoglobulins and is abundant in mucosal secretions such as saliva, tears, and breast milk. It inhibits bacterial and viral adhesion to epithelial cells and directly neutralizes some toxins. It is a very weak complement activating antibody. IgM comprises about 10% of the total immunoglobulins. It is the major immunoglobulin produced during the primary antibody response on first exposure to an antigen. Therefore, the presence of IgM generally indicates a current or recent infection. It is a potent activator of the complement system and a powerful agglutinating antibody, thus is efficient in neutralization of antigens. IgD is secreted in small amounts and seems to function mainly as a cell surface receptor for antigens. And IgE primarily defends against parasitic infections and is also involved in immediate hypersensitivity reactions and atopy. IgE binds to mast cells and basophil surface receptors with very high affinity. Antigen binding to the IgE then triggers release of inflammatory substances from these cells, such as histamine, resulting in some of the clinical manifestations of an allergic reaction.
Thanks, Maddi. So as you mentioned, the IgM indicates the presence of active or acute infection. And it's very important from the clinical point of view to find out when you order serology, if you detect IgG presence, that means it's either a chronic infection or patient is already immune to that particular infection, either via vaccine or innate immunity. If you look at the IgM, for example, Hep B IgM, that means the infection is active infection. Now, let's consider a scenario which happens in hospital not frequently, that is the contrast reaction in radiology department. And that can be classified as a hypersensitivity reaction. So can you please briefly describe the pathophysiological processes underlying the hypersensitivity reactions and give an example of each?
So hypersensitivity is an exaggerated or inappropriate immune response that results in tissue damage. There are 4 types of hypersensitivity reactions: type 1 or immediate, type 2 or cytotoxic, type 3, which is immune complex mediated, and type 4, which is delayed. So type 1 is the immediate or classic hypersensitivity reaction. IgE is synthesized by B cells on first exposure to an antigen and binds to the mast cells and basophils by the high affinity Fc receptors. On repeated introduction, the antigen binds IgE on these sensitized mast cells, resulting in degranulation. This results in the release of vasoactive substances, including histamine, heparin, and platelet activating factor, which causes vasodilatation, smooth muscle constriction, and increased vascular permeability. This may result in systemic or local effects. Examples of immediate hypersensitivity reactions include food or insect allergies. Type 2 or cytotoxic hypersensitive reactions are antibody-mediated cytotoxic reactions, which involve IgG or IgM antibodies binding to a cell surface. This results in complement activation, causing mast cell activation, neutrophil recruitment, and cell lysis via complement membrane attack complex and phagocytosis. Examples include transfusion reactions, hemolytic disease of the newborn, and myasthenia gravis. Type 3, or immune complex hypersensitivity reaction, is an immune complex mediated reaction resulting in deposition of antigen-antibody complexes in host tissue. This also activates complement and neutrophils, resulting in tissue damage. The immune complexes can also be deposited in the walls of arterioles, resulting in vasculitis. Examples of type 3 hypersensitive reactions include rheumatoid arthritis, SLE, polyarteritis nodosa, and post-streptococcal glomerulonephritis. And finally, type 4 or delayed hypersensitivity reactions is cell-mediated without complement activation and without the formation of antibodies. The antigen is presented to T cells, which then become sensitized and proliferate. The T cells release cytokines, which attract macrophages and lead to local inflammation. This response can take hours to days. After prolonged antigenic stimulation, the macrophages can then fuse to form giant cells. Examples include contact dermatitis and tuberculosis.
That's great. And I guess in ICU, we see all those 4 different hypersensitivity reactions in variety of patients. So it's very important to have a clear understanding of those hypersensitivity reactions, how they work, and what is kind of treatment for each one of them. Now, we're coming to the last part of our podcast, which is always the most exciting part for listeners and you. That is your joke. Obviously, it's not exciting for me, but I have to go through this exercise every single time. So, yeah, do you have a joke for us this time?
Yes, as always. So, how do you cure an allergy to gluten?
Oh my gosh, Maddi, that's very simple, isn't it? We just give steroids to everyone. So just give steroids.
No, by using the antidote.
Oh. Oh my gosh, Maddi. I think COVID has triggered your imagination even more, looks like. So obviously your jokes are a bit better after COVID isolation. Thanks, Maddi. And thanks, listeners. So this is the end of our today's episode. We'll be back in months' time with another episode of ICU Physiology. Till then, goodbye and have a nice time.
Thanks for listening. See you next time.